Thank you. Yes, as indicated, I'm Tim Bertram, the CEO of inRegen. inRegen is a clinical stage biotechnology company that is developing an autologous cell therapy for the treatment of chronic kidney disease. We actually envision the ability to eliminate the need for transplantation and dialysis. That is our vision. Whether we achieve that or not, I'll share with you some of our data, and you'll see the target on that. One of the things that is relevant to us is to know how to advance. We are a privately funded biotechnology company with our headquarters located in Camana Bay in Grand Cayman. The company was acquired 2.5 to 3 years ago, also privately funded at the time, and broken up into three parts. Our manufacturing component, which is located in North Carolina, was kept in North Carolina. Our clinical component, which is also located in North Carolina, was maintained there, and then the corporate headquarters was moved to Camana Bay. What's been interesting about this whole experience is running a biotechnology company as a virtual network internationally. What we've been able to do is establish some very, very strong partnerships with a number of different consultants, sponsors, supporters that have really allowed us to continue to advance the technology very effectively in the clinics from the phase 1. We currently have a 13-site clinical trial, which is growing to 20. If the product is successful, we believe that we can develop this through registration with 50 clinical sites. The clinical sites are chosen as major centers of kidney disease and really span the globe. We work with primarily in the U.S., wanting to go from coast to coast, but we also have been involved with regulators in Europe, including very close working relationships, particularly in the phase 1 at the Karolinska Institute with the Medicinal Products Agency. Because we have a footprint, corporate headquarters in the Cayman Islands, we've opened up a clinical center in the Cayman Islands. It's interesting for us to have that as one of our sites because uniquely the Caribbean area has a incidence of diabetic kidney disease, which is the major cause of end-stage renal disease, of about 2 times that found in Europe or in the United States. This has allowed us then to continue to contemplate how we can actually go forward in a very, very large market. Currently, our clinical trials have been conducted focusing on diabetic kidney disease. Diabetic kidney disease was chosen in collaboration with the regulators. We originally wanted to conduct an all-comers, but with 80% of the market found in diabetics who also happen to have hypertension, what it allowed us to do was go toward a $50 billion a year market that currently is really not being resolved in any other means. What was interesting for us, particularly if you look at that smaller graph, which you can barely read, that reflects CMS spending on end-stage renal disease. There's a growth of that of between 3 and 4%. That's where the $50 to $60 billion per year is obtained. There's a high level of interest by CMS in our product. There has not been with the FDA, at least in CBER, interactions between CMS and the FDA, and we're looking to pioneer a pilot program with them in hopes that we can bring in the CMS as a parallel review. We currently do have fast-track status. We've been suggested to go for RMAT, but we want to complete the phase 2 trial before we do that, and I'll share that data with you shortly. Because of the concentration of kidney disease within the diabetic population, that's obviously our major focus as an indication to get forward into a commercial status, if possible, as quickly as possible. What we're focusing on first is an indication of delay of dialysis. This would be a single injection of the cell therapy into a single kidney. That's how our phase 1 study was conducted. And what we learned in our phase 1 study is that the majority of patients actually can be dialysis-free for approximately 4.5 years. We did have one patient that went on dialysis from the phase 1 trial, and that individual started dialysis after 3.5 years. We've also decided to look at non-diabetic patients. The comorbidities found in that population are substantial. It's a good target population, particularly given the response of our kidney therapy. But we also would like to get to younger patient populations, so the congenital anomalies of the kidney and urinary tract is another target that we're moving toward. And that protocol was just approved by the FDA last month. In addition to that, one of the things that we think is quite significant is there are a number of diseases such as polycystic kidney disease and Fabry's and other genetic disorders that we believe that we can also go after with this therapy with some genetic modifications. Ultimately, and we do have projects ongoing to look to see if we can induce a tolerance/genetic modification that would allow for a universal donor. As I indicated, this is an autologous product going toward a universal donor. If we can see the same effects, that would be done. I wanted to review very quickly for you some of the data that we've seen in our phase 2 trial. I alluded to the phase 1. Phase 1 responses were quite profound. Substantial reduction in the decline of renal function, and it was sustained for approximately 4 years, and a number of the patients are still dialysis-free. In the phase 2, we actually saw a number of interesting effects that were exciting for us. I illustrate here 2 of the endpoints. The statin C, like creatinine, is part of the eGFR, or glomerular filtration rate calculation. And the phosphorus, hyperphosphatemia, secondary hyperparathyroidism is a common consequence in CKD. What we learned in our animal studies, we saw in our phase 1 trial, we're beginning to see in our phase 2 trial is that we don't just improve or stabilize their eGFR, but we also see the anemia of chronic kidney disease go away. We see the electrolyte abnormalities of CKD go away. The hypertension is reduced. Therapies can be reduced, if not eliminated. So there are a number of effects that we're seeing, not just simply improved filtration. What you can see here is we look to be seeing a stabilization, not a reduction in the decline. For those interested in the numbers, you can see that there's still a slight change, increase in the serum creatinine and cystatin C. We want to explore this as part of the repeat dosing paradigm that I talked about, where we'd actually prevent. This is, again, a single dose in a single kidney done in a diabetic patient population that had CKD 3B4 and is a randomized 1-to-1. So the solid line represents the control group, the blue line represents the treated group, the shaded area is the 95% confidence interval, and you can see where it says 0, that's the injection point of the product into the patients going forward. Just lastly, and I think I've run over, I'm sorry about that, but none of you I saw fall asleep. So this is just some highlights I'll leave up. No need to answer. Happy to answer any questions. Yep.
Great, Tim. So I guess the first thing I want to ask you is, for your autologous therapy, which types of cells are you trying to isolate and then re-inject?
Yeah, I'd love to say this was our brilliance and all the great scientific insights. This research actually was started 14 years ago. It was what I like to call a Thomas Edison approach when he was looking for the tungsten filament. He didn't know what he was looking for, and he just was pushing through all kinds of different cells that we had taken from the kidney. We actually started by taking the kidney apart and putting back into to see if there were effects, and it was simply a grunt through looking at the effects. We used 4 animal models, all that would die if untreated. So if the effect wasn't present, animals would die. If the effect was present, we then started adding back cells until we got the right combination. So that's how we created it. No brilliance, just a lot of hard work.
So is there a specific number of types of cells that you're re-injecting?
Yeah, we're actually, this is and some of you may be aware, for those of you that follow this literature, Ben Humphreys of the Harvard Stem Cell Institute, now at WashU, published a paper on the 6-2 population that was viewed to be the repair cell. It's found in the S3 segment of the proximal convoluted tubule, so very up near the glomerulus. It turns out, as we began to piece things back together, that it was that population that we found to be most effective in giving us the greatest effects. So we just gradually figured out how to manufacture that. Most of the problems with that, and we couldn't figure out why it hadn't been found before, but the cells are highly fastidious. They don't like to grow. They need to have contact. Too much contact, they undergo an apoptotic process. Not enough, they undergo an apoptotic process. They're a very delicate cell type, so a lot of effort went into the manufacturing, the formulation, and the product. The product is shipped fresh. The cells, we can freeze them in our laboratories, but in the hospital setting, when the physician's actually injecting them, we find that they're not as attentive to the thawing process. So if we ship fresh, we get a very consistent response in the patients from that cell population.
And what's the turnaround time from sourcing the cells to re-injecting?
Yeah, we run a standard biopsy. We take 2 through passes. For anyone familiar with this, you can have up to 5 through passes, the kidney cortex. We don't need the glomeruli since it's proximal convoluted tubules. So we take 2 of those passes. 4 weeks later, we have a product ready to go. In the randomized trial, we have stop places along the manufacturing process. We can, with those 2 biopsies, make a lifetime supply of cells for patient to get into our re-injections. So what we'll do with that then is we freeze them. If the patients go into the deferred group, the cells are stopped at stage 1, frozen away until the year and a half later they come back and are re-injected. For the active group, we immediately go into manufacturing 4 weeks, and the cells are injected back into the patients.
Okay, and what have you learned from the current phase 2 study about either the right type of patient, kidney disease with diabetes, but even within that universe? Is there a good candidate, a poor candidate? What have you learned there?
Yeah, this is an interesting question. We've studied that extensively, and both in Europe and in the U.S., trying to look through that. From a clinical trial development progress, the rapid progressing patients, those patients that are declining rapidly, are the best patients that we could want. Quick, easy responses, we can identify them. Those patients that are and the more severely affected also, I didn't show data here, but more severely affected patients, we've actually shown we can double their kidney function as measured by cystatin C or eGFR. If they're not so severely affected, say running around 50 ml per minute in their eGFR, what we see is just a prolonged stabilization of that. And so back to your question, the selection of the patients, I think for the conduct of a clinical trial, rapidly progressing, more severely involved, at least for our technology, are the best patients.
Okay, and what else have you learned, more on your end in terms of staging, scheduling? Those seem to be critical issues for you as well.
Yeah. So one of the things that we've done is to control the staging and the presentation, because there is a biopsy involved, and because there is an intervention. We work very, very closely with the interventionalist. And by working then and bringing the patients in with their regular schedule with the nephrologist, then the scheduling with the interventionalist, and that has really smoothed a lot of the interactions, because that's where it all counts. They both take this, they source it, and they inject it.
And what is the total number of patients you're looking to evaluate in the study?
Yeah, so we've only got 10. We recruited 6 last week, so we'll have 16 in this. They've just undergoing a biopsy. We're after 48. This is a 1-to-1 randomization, 24 in each. We'll probably do an open label extension trial to just carry this forward if it looks like it's worth doing. We'd like to do an end of phase 2 meeting with the FDA. We have, as I said, we had fast track. The FDA has been very, very good to us and with us. We're debating whether we do the RMAT just to spend the resources on it. We may, but if the end of phase 2, if they give us a combined phase 2-3, I think that's what we'll try to look to. We've calculated that we can probably do a phase 2-3, getting statistical significance with an 80% power and p.05. We can probably run 300 patients in a 3-to-1 randomization and see a positive outcome if the data stays consistent.
And do you have a sense of what's good enough in terms of the efficacy?
Yeah.
Stabilization, obviously, you're seeing some of that, but in terms of prolonging more acute or serious intervention, what's your guess at this point?
Yeah, exceptionally good question. I wish I knew the answer to that. Our view is if we can give 4, and CMS has told us this as well, if we can give 4 years of dialysis-free living, for them it's a score. What does that mean, though, in terms of the staging and where it's at? I think it's an excellent question. What we're looking at is probably setting some kind of a of a stabilization and then monitoring for a decline and then redosing. And kind of a stage gate approach is how we're thinking about the redosing for the prevention.
Okay, well, very good. Thanks so much.